Day, everybody. So I can't be here. Today you're going to be learning about lipids, another macromolecule. Yesterday was carbohydrates. Today is lipids. Let's go ahead and set your Cornell notes up and give you a second to do that. Here's our target. Students know most macromolecules, proteins, fats, and carbohydrates in cells are synthesized from small collection of simple parts. So same one as yesterday, just we're doing the same thing, only concentrating on fats. You'll see how the small parts of fats make bigger things of fats. So go ahead, put that at the top of your page. Make sure you got your heading, left side, right side. Go ahead and do that right now. All right, so one important thing with lipids is they store a lot of energy. You can see Andy Reid here, the coach of the Kansas City Chiefs. He has stored a lot of energy. And fats that we normally think of, like what you see in this nice ribeye, those are what we call triglycerides. And fats are made from glycerol, which is the glyceride part of triglyceride, and fatty acids. So the two small parts are glycerol and fatty acids. Put them together, you get a triglyceride. Like when we're talking about carbohydrates, you had two monomers come together to make a disaccharide. So that's the similarity there. One thing about lipids is that they're hydrophobic. That means that they don't like water. You can see in our little glass right here, the water is separated from the oil. So oil is another type of fat. All right, so here's a little picture explaining triglyceride. You have the glycerol molecule, which is this portion right here, and it's bonded to three different fatty acid tails. And the fatty acid tails are a series of carbon molecules that are surrounded by hydrogens. And so each one of those bonds is stored energy, and when we break those bonds, it releases the energy. They are non-polar, and remember water is polar, so that means it's not going to mix like with water, just like we saw right back here. It doesn't mix because it's non-polar and water is polar. And again, hydrophobic means it is afraid of water. It doesn't mix. So fatty acids linked to the glycerol by dehydration synthesis. Same like we saw before. You lose the hydrogen and the hydroxide ion there, and that's going to turn into water. HOH, H2O, that's water. When that happens, we join our molecule. And that takes energy to join molecules. It's saturated when all those bonds around the carbon, all of these guys, each carbon has two hydrogens on it, except for the last one here, which has three. So it has as many hydrogens around it as it can possibly have. That means saturated. So some examples of that are things like butter. So animal fats are saturated fats. They're solid at room temperature. So you can put butter on there. Go ahead, take a moment, and try and think of, a couple different saturated fats. So go ahead, push pause for 30 seconds. Now, go ahead and tell your neighbor, see if you came up with any that are similar. And then push pause here while we have about a minute. All right, and then go ahead and share with your instructor what are some saturated fats. Push pause while you're doing that. All right, so now hopefully you've got things like bacon, that fat in the ribeye, lard, those are all some saturated fats. And think about it, the bacon, when you take it out of the fridge and leave it out, it has that nice fat on there. And not until you heat it up in the skillet does it turn into liquid. So we have also unsaturated fats. These are ones that have at least one double bond. So right here, the double bond. And you notice here we have our saturated fatty acids, and all of a sudden there's this double bond right here, and it kinks. So that's going to be important here. That little kink makes it harder to pack these together. When they're packed together, they line up nice and neat, and you can make a solid. When they don't pack together, it's going to end up giving you a liquid. Plants tend to make lots of unsaturated fats, lots of oils. They're liquid at room temperature. They're more fluid, and that's because you can't stack them together because they have these little kinks. So now, go ahead and talk with your neighbors. Give yourselves a couple minutes. Try and answer these questions. What are two small parts that make up a macromolecule of fat? What is an example of a saturated fat? And what is the difference between a saturated and unsaturated fat? So talk with your neighbors about that. Go ahead and push pause and then share the results with your instructor, what you have. And then I'll go ahead and tell you some answers that I thought of. So two small parts would be glycerol and fatty acids, and in fact we need 3 fatty acids for each of those glycerol molecules to make a triglyceride. An example of something that has saturated fat would be butter or lard, bacon, I don't know, there's lots of things. The difference between saturated and unsaturated fat, you could say saturated fats are solid at room temperature, unsaturated are liquid. You could say saturated fats are, they have no double bonds, they're all the carbons have the maximum amount of hydrogens around them, where unsaturated fats have those kinks in them, those double bonds. And saturated fats are less healthy for you than unsaturated fats, because what will happen is they'll clog up in those arteries because they stack nice and easily, where the unsaturated fats move around a little bit more. Phospholipids are another really important type. It's probably more important that you pay attention to this than any other thing, because we're going to talk about this with our plasma membrane next week. So phospholipids, again, we have fatty acids right here. We have 2 fatty acids and a polar head. So this is a little bit different. The triglycerides, they are all nonpolar. For our phospholipids, you have a nonpolar portion, which doesn't mix with water, and a polar portion. And if you notice, it says hydrophilic, like. All right, so these like water, the nonpolar don't. They're hydrophobic. And they, like I said, they make up our cell boundary. So all these little balls with the 2 little tails, those are our phospholipids. And there's 2 layers here. So we'll learn that this plasma membrane is called the phospholipid bilayer. All right, so we can illustrate a phospholipid. I want you to go ahead and try that. And it doesn't have to be anything special. So go ahead and put that into your notes. And then talk about what's the difference between a phospholipid and triglyceride. So push pause, give a few minutes. All right, I'm back. So hopefully you have a nice little phospholipid drawn. Actually, let me put the phospholipid back up for you. All right, now that you've got your phospholipid drawn, a difference between a phospholipid and a triglyceride. There's many. Triglycerides are all nonpolar, so they don't mix, where phospholipids are what's called amphipathic. They kind of one half mixes, one doesn't, because it's got a polar head and a nonpolar tail. The phospholipids have 2 fatty acids instead of 3. Triglycerides are used for energy storage, whereas phospholipids are used in our plasma membrane and lots of different membranes. Right? There's more than just phospholipids and triglycerides. We have steroids. These are in ring structures. Again, we have lots of CHs. These are used for lots of different things. Some steroids, like cholesterol, are used in our plasma membrane back here. All right, so they help us keep the membrane fluid and moving. Some steroids are hormones like androgens, estrogens, progesterones. These are hormones. They help us create different things to run and regulate our body. We have anabolic steroids. Remember, anabolic means getting bigger, building things. So like Lou Ferrigno here, known for using steroids. Not a good thing, but something to help you remember. We also have cholesterol, like I mentioned before. Some cholesterol is good, some bad. Sometimes it clogs up the arteries. There's different types of cholesterol. All right. And then what I want you to go ahead and do now is, ooh, that looks good. Go ahead and take a moment to pause and write a summary. And then your teacher's going to go ahead and call on you to hear some of your summaries and some of your questions. All right. Thank you very much.